Okay, Ben, thank you very much for the kind introduction. Thank you for the invitation, and thank you very much for organizing this. I think this is a fantastic initiative. Um, so I'm gonna show today some of my postdoctoral research about studying the biophysics of protein surface interaction. And I first wanted to start by explaining why we care about protein surface interactions, why we think are important. Interactions of proteins with biological surfaces occur in many important physiological processes that go from intermolecular recognition, cellular communication, all the way to, for example, something that's very important nowadays, viral infection. Here, for example, it's an image of HIV protein inserting into a cell membrane during an infection process. So interaction of proteins with biological membranes are occurring in many physiological processes, also in many pathological processes. For example, detrimental interactions of proteins with cell membranes can induce irreversible misfolding and aggregation that triggers neurodegenerative diseases. So a better understanding of protein interactions with surfaces, it will help us understand biological processes, whether physiologically important or pathogenic. We also care about the interactions of proteins with artificial surfaces. Artificial surfaces have very different chemical composition, very different chemical properties than biological surfaces. And very often, whereas proteins remain functional and folded on biological membranes, they irreversibly adhere and aggregate onto artificial surfaces. And that is very often a limiting factor when we try to use proteins in biotechnology. In the context of our lab, the Plaxco lab, we're trying to develop new tools for personalized. And in my particular case, I'm interested in developing protein-based electrochemical sensors. So being able to understand these interactions will help us implement proteins in biotechnologies and biosensors. So the motivation, again, it's a better understanding of biological, physiological, and pathological processes, better ability to implement proteins in biotechnologies. And there is also a more fundamental reason, which is understanding how different the physics of proteins on surfaces are from the physics of proteins in bulk solution. We expect that there will be different thermodynamic contributions when proteins are on surfaces that we don't typically see when we study proteins in bulk solution. So for example, if you can think that the protein, when it's interacting with the surface, the excluded volume of the surface is going to impose a restriction in the conformational entropy of the protein. There's going to be some conformations that are not going to be allowed because of that volume that the surface is occupying. So that is going to affect preferentially the unfolded state of the protein because it's more disordered. So it's going to have more conformations that are going to be disallowed. So this contribution is preferentially going to restrict the conformational entropy of the unfolded state, and therefore it's going to stabilize the folded functional conformation of the protein. Interactions with the surface, in contrast, are likely going to be destabilizing precisely because the higher flexibility of the unfolded state is going to allow it to better fulfill the attractive interactions with the surface while at the same time avoid repulsive interactions. So again, this is going to favor the unfolded state, so it's going to be destabilizing. And then, of course, the balance between these two opposing contributions and perhaps other contributions, it's going to be heavily dependent on the details of the system. For example, the amino acid composition of the protein, the chemical composition of the surface, or the topology of the interaction. So we want to be able to measure we're going to be able to quantify all these thermodynamic contributions so we can understand better the biophysics of protein surface interactions. How can we do that? Well, our approach to do that is to first measure the free energy, the stability of the protein when it's in solution. There we're going to measure the folding free energy, the stability of the protein when it's attached to the surface. And then the difference between the two it's going to inform us on the extent to which interaction with the surface it's stabilizing or destabilizing the protein, which we use as a proxy to whether interaction with the surface it's favored or disfavored. So this difference between these two stabilities is going to give us the interaction-free energy. It's going to give us that quantitative measure that we want to obtain to be able to understand in a systematic way protein surface interactions. So it all boils down to measuring the stability of proteins. As I've been warned that the audience today will be a bit heterogeneous, I wanted to take a little detour. I'm gonna spend a couple minutes explaining how do we typically measure protein stability. So when we refer to protein stability, we're talking about in this case, the unfolding free energy. So that depends on the equilibrium constant, which depends on the ratio of the folded and the unfolded populations. Normally for stable proteins, the typical stabilities, for example, of the proteins that I'm going to talk today, of 20 kilojoules per mole, for example, that means that given the exponential dependence of the folded population with the folding free energy, that means that we're going to have more than 99.9% of the protein is going to be folded, and 0.03% it's going to be unfolded. So it's very difficult to measure protein stability on these conditions because we are not able to accurately measure the folded and the unfolded state, because the population of the unfolded state is nearly negligible, right? So there is a way that we can measure those stabilities, which is doing chemical denaturation experiments. We know that the folding free energy of the protein depends linearly on the denaturant concentration. So then what we do is we measure the folded and the unfolded population as a function of denaturant. So we keep adding denaturant, the folding free energy decreases linearly, and then when we reach the point where the folding free energy is 0, we have 50% of the folded and then the unfolded state. So here we can very accurately measure the populations of both the folded and the unfolded state. So it's in this region around here when we can measure populations, we can determine accurately the folding free energy. And as we know that the dependence is linear, we just extrapolate back to 0 denaturant, and that's what's giving us the stability of the protein. So this is how we do it. We have a technique that allows us to sample the folded and the unfolded populations, and we add denaturant, we measure this unfolding transition here, and we can obtain our folding free energy. So it all boils down to having a spectroscopic tool that it's able to measure the folded and the unfolded population. And that has been done for decades already in bulk solution. We take, for example, optical methods like circular dichroism, which are sensitive to the amount of secondary structure of the protein. So when the protein is folded, it's going to be composed mostly of alpha helix and beta sheets. So we're going to have a negative circular dichroism signal. And then as the protein unfolds, it's going to go to the random call that has a much closer to 0 circular dichroism signal. This is a real experiment. Experimental data. We start in the absence of denaturant, the protein has a negative ellipticity, so it's folded. And then as we add denaturant, we start losing ellipticity until we reach the unfolded state, which is very close to 0. So if we keep tracking the ellipticity of the circular dichroism signal at a constant wavelength, what we see is that first we have a baseline here, which corresponds to the folded state. Then we have this cooperative unfolding transition. And then finally, we have the baseline of the unfolded state. And from this denaturation, we can extract the folding free energy of our protein. So we can do this in solution with, for example, circular dichroism, because sensitive to the secondary structure of the protein. We need to come up with a technique by which we can monitor the folded and the unfolded populations of proteins on surfaces. And there is not such technique. So we had to come up with an approach by which to measure that. So for our studies, we used Protein-L. Protein, it's a very well characterized single domain, two-state folding protein. We made a protein variant in which we introduced a cysteine group, and then we mutated, we substituted all the lysines into arginines. In this case, we use those mutations because they keep the same net charge as the wild-type protein. And we remove all the lysines because we want to remove all the amine groups except for one, which is the N-terminal amine. So now we have two orthogonal unique chemical groups, the thiol in the cysteine and the amine group at the N-terminus, that we can use to modify our protein and be able to measure its stability on the surface. So first, we're gonna conjugate this thiol group with a methylene blue. It's our Redox Reporter. And then we modify our N-terminal amine group with an thiolated alkane that we're going to use as an anchoring molecule to deposit our protein in the surface of a gold electrode. We then coat the gold electrode with a self-assembled monolayer of mercaptohexanol that's passivating the gold surface, and it's providing us with a homogeneous surface, which is hydroxyl coated. It's compatible with biomolecules. And in this configuration, when the protein is in the folded state, the methylene blue is held close to the surface. So we have a fast rate of electron transfer. Then as some folding occurs, that's going to pull the methylene blue farther away from the surface, which is going to slow down electron transfer. So we've managed to connect the conformations of our protein with a property that we can measure, that we can detect, which is the rate of electron transfer. We can use electrochemical techniques that are sensitive to this electron transfer rate to monitor the conformation of our protein. We're using the square wave voltammetry. Here, we can see that the rate of electron transfer of the folded state it's faster. So we have a faster decay here than the unfolded state. And then if we measure electron square wave voltammetry at 600 Hertz, we're sampling this initial phase of the decay. So we have a high peak current in the folded state. And then as we add the denaturant, we are going to see a decrease in the peak current corresponding to the unfolding of the protein. So if we monitor the peak current of our protein as a function of denaturant, we're going to reproduce the expected two-state unfolding transition that, if you remember from our theoretical picture early on, we are expecting for protein L. So first we have a folded baseline. This baseline here corresponds to the folded state. Then as we approach the point where the free energy is zero, we are seeing this cooperative unfolding transition. And then finally, we have this unfolded baseline here. So this is an unfolding, this is a typical sigmoid corresponding to a two-state protein unfolding. It is reversible, which means we're measuring at equilibrium. So now we can analyze, we can apply the equations that describe the two-state folding equilibrium of proteins, and we can extract the folding free energy of the protein when it's attached to the surface. And that's giving us this value. It's 27 kilojoules per mole. And now, by comparing this number to the stability of the protein measured by the same technique in bulk solution, we can evaluate whether interaction with the surface is stabilizing or destabilizing for this protein. What we see is that the stability on the surface is higher than in solution. So interaction of protein L with this hydroxyl terminated surface, it's stabilizing by 6 kilojoules per mole. Now I'm telling you, but why should you believe me? How do we know that this is a real effect? We obviously run some controls, and I'm just going to briefly show one of them. We know, for example, that cosolutes affect the stability of proteins. For example, we know that sodium sulfate, it's a stabilizing cosolute. So we tested sodium sulfate on the effect of sodium sulfate on protein L when it's on the surface and when it's in solution. And in both cases, we see a stabilization. And the extent of that stabilization is the same for both of them. And the same thing happens for denaturant urea and guanidinium chloride. So we're pretty confident that what we're seeing is real. And we are seeing a stabilization. So where is this stabilization coming from? This is the interesting question. We're observing an effect Where is the effect coming from? If you remember, we were talking at the beginning of what contributions we were expecting. And we were talking about a stabilizing contribution that we were expecting due to the excluded volume effect of the surface that will be restricting the conformational entropy of the unfolded state. That will therefore destabilize preferentially the unfolded state, and that could explain that stabilization that we see when the protein is interacting with the surface. However, the data that we are measuring is thermodynamics. So it's devoid of mechanism. We only measure the start and the end point, but we don't really know what's going on in between. This is kind of like a black box. We were expecting that other contributions would also be important. We were expecting interactions to be important, and in particular, we expect that electrostatic interactions are going to be important. Why is that? Well, because the surface that we employ, the hydroxyl terminated surface that we employ, at the experimental conditions that we employ, the potential of methane blue, ionic strength, pH, etc., it's expected to be slightly negatively charged. And the protein that we're using, proteinal, it's also slightly negatively charged. So we have charged amino acids, 6 positively charged, 10 negatively charged, and then the net charge of the protein, it's -4. So in order to evaluate to if and to what extent electrostatic interactions are important, we have designed other variants of proteinal with a different charge composition. We have made another variant which is more negatively charged. We have managed to remove all the positively charged of residues on the protein, so we're left only with negative charge. So that is the most extreme negatively charged variant that we have. We've also built neutral variant in which the number of positively charged residues and negatively charged residues is the same, so the net charge is 0, and also to evaluate whether it's just an effect of net charge or also a specific charge pattern, charge composition. We made a second neutral variant, but with an overall different total number of residues. For each one of these variants, we're measuring the interaction free energy with the surface. So we measure the stability on the surface, we measure the stability in solution, and we compare the two. This is the one that I already showed you, it's the parent proteinal. It's stabilized by surface attachment by about 6 kilojoules per mole. When we measure the more negatively charged variant, we see that stabilization, it's also stabilized, but to a lesser extent by the surface. And then the most negatively charged variant that we've tried, the homogeneously negatively charged variant, it's not stabilized at all by the surface. And then if we go on the opposite direction, the neutral charge variant that we measure, it's more stabilized by the surface than the parent proteinal. Now, surprisingly, when we measure the second neutral charge variant, we find that it's also stabilized, but not as much as the other neutral charge variant. So overall, there are two important things here. One, it seems that details like the charge composition matter. And second, there seems to be an overall trend in which the more negatively charged proteins are less stabilized by the surface, which intuitively makes sense. You have a surface that it's supposedly negatively charged. If you put something that it's also negatively charged on top of it, it's going to be less stable. We wanted to see whether this is general for other proteins, so we measure another similar protein, Finesse H3 domain. It's a protein that it's similar in size to protein L, slightly smaller, but it's more negatively charged. And then qualitatively, it fits the trend. It's more negatively charged, so it's less stabilized by surface attachment. Well, it's not stabilized at all by surface attachment. So at least qualitatively, it fits the overall picture. Then also, the parameters that we can extract from this linear correlation, they also make sense with our physical expectations. Let's look, for example, at the intercept with 0 here. That value should be informing us on the effect of the surface on protein stability when electrostatic interactions are minimal. So it should be, in principle, our best approximation to the excluded volume effects, or at least where we can get rid of most of the, or minimize the electrostatic interactions. So the experimental value that we get, 9 kilojoules per mole, it's quite well in line with theoretical and computational predictions of how much this effect should account for. For example, Kendall and others calculated a 12 kilojoule per mole stabilization for a 200 residue Gaussian chain. Now, this makes perfect sense with our data because if you have a long, well, I should say, protein it's 64 residues. So this is more than 3 times longer. So it makes sense that if you have a longer chain, you're going to have more conformations restricted. So you're going to have a higher entropic penalty. So this is in line, at least qualitatively, with our prediction. We can compare also with computational predictions on the entropic penalty due to this excluded volume effect on a self-avoiding random polymer chain of length equivalent to that of our protein. And the value that they get is 8.6 kilojoules per mole, which is really close to the 9 kilojoules per mole value that we obtain. So it seems that we are seeing, to some extent, this excluded volume stabilization. Then we can look at the slope of this dependence too. This slope is about 1 kilojoule per mole per charge. This would account for the number of, for the energetic cost of bringing one charged unit close to the surface. Now we can calculate, since we know the charge at the surface of the protein and we know our solvent conditions of ionic strengths, etc., we can calculate the potential of the surface as a function of the distance from the surface. The 1 kilojoule per mole value on free energy that we obtain, it's equivalent to bringing one charged unit from the bulk to within 4 angstroms for the surface. And this makes sense with the physical distances of our protein. Protein radius of gyrations is about 15 angstroms, so this makes sense with, and the unfolded state, it's about 30 angstroms. So I think this is consistent with having charges on the polymer that are far from the surface in the unfolded state, and then when the protein folds, they get closer to the surface. So it seems that that we have a correlation that intuitively makes sense. We have a surface that's negatively charged, then we have a protein on top of it. The more negatively charged the protein is, the more destabilized it is by the surface. You have a constant or general stabilizing effect due to excluded volume that matches theoretical expectations, and the extent to which adding more negative charges are destabilizing, it also matches theoretical expectations of the cost of bringing a charge close to that surface that we're employing. So this is what we understand. This is the interpretation we feel comfortable with. However, we also looked at the correlation of this interaction-free energy with other properties of the sequence. And we found an even better correlation with the total number of positive charges, which we're still trying to understand exactly what this means. So since this is an informal venue, I figure it would be appropriate to show that sometimes you don't really understand everything that you get out of your experiments. We're not certain what this means, because if this was the case, you could talk about attractive interactions between positive charges and a negative surface, but then you would expect a similar but opposite correlation with the number of negative charges, which we don't see. It might be related to the fact that the only positive charges that we can use are arginines, because remember, we want to have a unique N-terminal amine and unique amine group, so we get rid of all the lysines. So perhaps what we're seeing is a specific effect of the 1-edenium moiety of the arginine with the surface. We're not 100% sure of what that means. Overall, anyway, I think our data support some of our initial hypotheses, which is that interaction of protein with the surface will have some excluded volume contributions that destabilize the unfolded state. And then we have also charge residue composition being important in modulated to the extent to which this hard repulsion with the surface is stabilizing the protein. So this is already telling us about how different the physics of proteins on surfaces are from the situation in bulk solution. We're seeing, for example, that excluded volume effects from a macroscopic boundary are stabilizing, which is an effect that we won't see typically in solution. But now, for example, what about intermolecular crowding on surfaces. All the data that I've shown you now, it's measured in what we call a dilute regime, in which the protein molecules that we deposit on the surface are on average far enough so they won't be having intermolecular interactions. Now we can control the amount of protein that we deposit on the surface, so we can control the packing density of proteins on the surface, and we can increase it to the point where the molecules are on average so close that they could have interactions. And when we do that, we see that that affects the denaturations that we measure. So we see that the denaturation broadens. When we fit it to our two-state model, we see that the M value, which is a measure of the stiffness of the transition, decreases. And we also see an apparent decrease in the stability of the protein. Now, these effects occur at the crowded regime, what we call the crowded regime. That's when the intermolecular distance in between two protein molecules, it's below the contour length. So the contour length would be the maximum length that the polymer could span if you were to stretch it. It's about 4 angstroms per amino acid. So how do we explain this? And we've seen similar effects, for example, for DNA when we measured DNA on surfaces. So the way we interpret this data, it's we have anti-cooperative effects that make the unfolding easier at the beginning and harder at the end. The way we interpret that, it's at the beginning, when you have a lot of folded molecules packed next to each other, you're forming a steric repulsion, which is destabilizing. And you're also promoting forming of intermolecular interactions, which will also favor the unfolded state. Now, as the denaturation progresses, every time more and more neighbors are going to be unfolded. And then you're going to reach the situation in which a folded protein is going to be surrounded by unfolded neighbors. So now the steric hindrance, it's opposing unfolding, because the protein, if it unfolds, it's going to compete with the space here. So it's going to, these unfolded neighbors are going to favor the protein to remain folded. So we have anti-cooperative effects. These intermolecular interactions are going to be dependent on the folded or unfolded population of your neighbors. And at the beginning, they're going to promote folding, but then at the end, they're going to prevent folding. So these are also different effects than what we would see in solution. These interactions with other biomolecules on the surface, these effects, these crowding effects, we won't see that in solution. And I think there is a very clear way of picturing why that's the case. When we have the proteins on the surface, they are immobilized, they are tethered to the surface. So even if you have those intermolecular interactions, you are preventing to form oligomers, high molecular weight oligomers that will aggregate and will irreversibly precipitate, which is not what you see in solution. And that's why if you calculate the concentration of protein that we obtain on the surface, it's above the solubility limit that we see in solution. So we can get concentrations on the surfaces much, much higher than you could ever see in solution. And that's why we can access new regimes in which we can observe different physics, different phenomena. I think this is pretty interesting from a fundamental point of view, how the physics of proteins on surfaces can be very different from those in solution. So this is an interesting observation of interactions of proteins with other biomolecules on the surface. What about how surfaces affect the way proteins interact with other biomolecules in solution? To explore that, we tested the effect of different co-solutes on protein stability. And I've talked a little bit about that before, but now we made a more systematic study. We studied more co-solutes. We studied some of Hofmeister ions that stabilize or destabilize the protein. We've also tested some organic osmolytes, which are well-known protein stabilizers, and we've tested also some denaturants. We've shown already that sodium sulfate, it's stabilizing the protein. This is what we typically see in solution. We're seeing that the denaturation curve shifts towards highest concentration of denaturant, so you need more denaturant to unfold your protein. So this is the effect of sodium sulfate stabilizing the protein, and we see pretty much the same effect on the surface. We see that the The M value, it's the slope of this dependence. It's the slope of dependence of the stability of the protein with the co-solute concentration, and it's the same for both of them. The offset is just because the stability of the protein, if you remember, it's higher on the surface than in solution. So sodium sulfate, same stabilizing effect. We then tested the effect of urea, denaturant, and what we see is that as we add more denaturant, the stability of the protein decreases in solution, and then we see the same on the surface, and the extent of that destabilization is the same. So again, the M value, it's the same. So as so far, this is expected. But then we tested sucrose. So sucrose is a well-known protein stabilizer in solution, as we can see here, for again shifting the denaturation curve. But when we tried on the surface, all these curves are pretty much overlapping with each other. So whereas we see a clear stabilizing effect of sucrose in solution, we don't see that stabilizing effect when on the surface. We tested another polyol, we tested the glycerol, and again, we see that stabilizing effect in solution, but we don't see that stabilizing effect on the surface. Those curves are overlapping with each other. So again, positive M value, stabilization on the solution, nothing on the surface. So overall, what we have is that ions like sulfate, denaturants like urea and wanidinium, they are behaving the same way in solution on the surface, whereas these two osmolytes, neutral polyols, sucrose and glycerol, are not. They are stabilizing in solution, but they have no effect on protein stability on the surface. So why polyols are failing to stabilize protein when these are attached to hydroxyl-coated surface? First, we want to see if this is a general effect. It could just be that it's something specific of proteanol. So we tested that on Finis H3, and in solution, we see that both, for example, sulfate and sucrose are equally stabilizing. No, sorry, not equally stabilizing, but both have a stabilizing. But when we test those on the surface, sulfate keeps stabilizing the protein to a similar extent to the one that we see in solution, but again, sucrose does not. So it seems that's a general effect in which polyols fail to stabilize proteins when these are deposited in a hydroxyl terminated surface. Now, does it depend on the chemistry of the surface? We have a hydroxyl terminated surface, and we're seeing a weird effect with the polyol co-solids. So it makes, I think this is a question that makes sense. So we tried different surface chemistry. We tried a carboxyl terminated surface. So instead of a monolayer of mercaptohexanol, we put on top of the electrode, we put a monolayer of mercaptohexanoic acid. And when we measure the effect of sucrose on the stability of the protein, we now see an effect. So we are increasing, we're pushing the denaturation curve towards higher concentrations of denaturant. So we're stabilizing the protein, but with sucrose is stabilizing the protein when it's on the surface. The extent of that stabilization, moreover, it's the same as the one that we see in solution. So by changing the monolayer, the surface chemistry, we are recovering the stabilizing effect that these polyol osmolytes have in solution. So the way we interpret this data is the following. The way these co-solutes affect protein stability, it's because they are preferentially excluded from the surface of the protein, and that preferential exclusion, it's more pronounced in the unfolded state. So they favor more the folded state. So the stabilizing effect of the co-solute arises because there is a differential partition between both states. Now, when these proteins are interacting with the surface, if the surface is strongly affecting the way the osmolytes partition in the vicinity of the surface, then you're always imposing the same concentration of co-solutes around your protein. So you no longer have a differential partition between the folded and the unfolded state, because all the partition coefficient, eventually, it only depends on the chemistry of the surface. And if you have hydroxyls that are strongly hydrogen bonding your surface, then you won't see that differential partition. when we add the carboxylates, we're probably not forming these interactions, and then we're not seeing that the surface it's imposing the partition of the osmolator on the protein, and then it's the protein again that can provoke this preferential exclusion, and then we see those stabilizing effects. So in conclusion, we have studied the biophysics of protein surface interactions. We've come up with a new experimental method by which we can measure the thermodynamics of these interactions. We've used it to explore what makes how different are the physics of proteins on surfaces from those in bulk solution, and we see that we have important excluded volume effects due to the surface that are restricting the unfolded state entropy. We're seeing also that the effect of charged amino acid composition is important in modulating this stability. We also see crowding effects because we can reach concentrations on surfaces way above the solubility limits that we could ever achieve in solution. And we also see that the surface chemistry can strongly affect the way proteins interact with other components of the system. And with that, I just want to conclude. I want to thank the group of the Plaxco Lab that were working in this project. Also, some of the former postdocs in the Plaxco Lab that now they have their own groups that's independent faculty, especially Netra Arroyo. Thank you for the recommendation for being here today. And then our collaborators from the John Shea Lab at UCSB and Dino Makarov at UT Austin, who are providing the computational and theoretical counterpart to the experimental work that we're doing. And that's it. Thank you very much all for your attention. I will take any questions. And I'll give it back to you, Ben.
That was a fantastic talk. I think there we go. That was a fantastic talk. I think I speak for everybody that's been here tonight when I say that was really fantastic. And that is reflected in the number of questions that you've got. So we'll just jump straight into it.
Okay.
So the first question is from Dr. Kotapalli. She wanted to know, how does the buffer composition affect these calculations, and how do you determine what buffer to use?
I guess you are referring to the electrostatic effects when we evaluate the effect of the slope.
I think that probably so, yeah.
Okay. So, yeah, so let me, if I can go back to that slide. So we're using phosphate buffer, and there is and some salt into it, but there is something more here. I think this is the one. Yeah. So the thing is that the denaturant that we use, it's an ionic denaturant. So we're using vanadium chloride. It's a salt. So at the concentrations at which we are obtaining our thermodynamic information, which is around the midpoint of the denaturation, we are at between 2 and 3 molar ionic strength. And that makes that decay in potential very steep. That said, I think this is also an interesting observation compared to solution. In solution, when you increase the ionic strength, you're making the reach of the potential much smaller than intermolecular distance. So two proteins that interact due to electrostatics or two biomolecules, they won't see each other because they're too far tumbling in solution. But here, the protein is confined to the surface. It cannot escape the potential. So no matter how small you do this, because eventually the protein is gonna, since it's attached, it's tethered here, it won't be able to escape. As to how do we choose our buffer working with proteins, phosphate buffer is a very common buffer. You need to add some salt. You need to have some background ionic strength in here. Yeah, we couldn't use urea to do these denaturations because urea is a less potent denaturant, and it gets very, very shallow transitions, and you cannot get enough folded and unfolded base. So for this protein, we're limited with vanadium chloride. But it's an important she raises an important aspect, which is that it would be nice to be able not to use an ionic denaturant, because then we could play around with this a bit more.
Yeah. Okay. All right, let's see. So I had a question. Does the size of the redox reporter affect the electrochemical measurement? And like basically, could you have one that was too big that would introduce so some other effect that you would have to account for? Maybe not necessarily like an electrostatic effect or something like that, but
yeah, that's a very important question. In this experiment here, this reference state, it's the protein in solution labeled with the methylene blue and the alkane thiol anchor that we use.
Mm-hmm.
So we wanted to control whether we had some effect of the methylene blue on the stability of the protein, which we did not. We might have some effect of the methylene blue on the interaction with the surface.
Mm-hmm.
Methylene blue, it's known that it can intercalate in many things, so it could be interacting with the surface. That's why we run those controls, for example, not those controls, but that's why we run experiments with different proteins, different net charge. So the methylene blue should be the same there. So if we have any effect, then it should not be due to the methylene blue.
Okay, interesting. So let's see. Kyle wanted to ask, have you tried electrochemical ATR, SEI, RAS? I don't know what that is. I know ATR is attenuated total reflectance. I do not know what the rest of that is.
Yeah, I'm not
familiar. An IR method, apparently.
We have not, but other people, I think they tried SPR.
Okay.
And the data that they got, it was frankly not very quantitative. Instead of having, as we see, what you would expect, which is a two-state unfolding in which you have a folded baseline, cooperative unfolding transition, and then an unfolded baseline. In their case, it was more like a straight line. So I guess we could apply different techniques. This is the one we use. Also, there is a practical motivation behind this work, which is that in our lab, we do electrochemical sensors, and that's the technique that we use to probe our sensors. So we're obviously biased into the technique that we use.
That makes sense.
Yeah.
Okay, that's interesting. Let's see. So Kyle also wanted to know if you could use fret to examine folding on a surface. He says he's just curious. It's because he knows it's been used in solution before.
Yeah, and some people, I guess the people doing single molecule experiments, I guess they could technically do that. It's just that the surface that you will have to use will be very, very different. So the surface that we use is the same that we use for when we deploy our aptamer-based sensors in vivo. So it's much more comparable if eventually we want to make protein sensors. In their case, I don't think I'm not sure if they can have the same type of surface, or if they could control, for example, the surface chemistry just by changing the monolayer the same way we do. But yeah, I guess technically it would be possible. They just didn't do it.
Okay. All right. So maybe possible, but not necessarily not going to give you the information you're interested in.
Yeah.
Okay. How does the SAM how does your monolayer affect the protein folding state?
We think we don't think it's causing any significant changes in the conformation of the protein, based on the evidence that we see that the effect of co-solutes on protein stability is the same. We also have a functional assay with a different protein for binding, and we see the same effect that you would expect in solution. So we don't expect that the conformation is changing significantly to the point that it will let's say, break the protein function. That said, we don't have high resolution structural information of our system. We cannot use NMR or X-ray crystallography here to measure the protein structure. So we don't have direct structural evidence. We have indirect evidence through all the thermodynamic parameters and functional assays and mutations and the effect of co-solutes that we see.
Okay. I guess that's a good check on it.
Mhm.
Okay. How would you correlate this with or how would you correlate your results with the Fulden volcano-shaped funnel model?
Uh, so so the way I interpret this would be, pull this the model slide one more time here. So again, I think what we're seeing is that we are probably increasing the free energy of the unfolded state. So we, I guess it depends. I guess it depends on what protein variant, right? Because in you have one general effect that it's making your unfolded state higher energy, so more disfavored.
Mm-hmm.
But then, depending on the electrostatics, you might be introducing some how to say, some gatekeepers, so you won't end up in some other alternate concentrations, alternate conformations um, that will decrease or that will deviate from the preferred pathway to the correct folded state.
Mm-hmm.
So we didn't really think about it in that sense yet, because we don't really have folding information here.
Okay.
But I guess I think we're affecting different conformations are affecting different parts of the folding trajectory, I
Okay.
would say.
She kind of followed up real quick with a question. What's the length or size of your protein? Um,
What's the what?
with the what's the length or the size of your protein in the completely neutral form?
So the radius of gyration of the folded protein, it's 15 Armstrong. The radius of gyration in the unfolded state, it's 30 Armstrong, I think.
Okay.
And then if you want to look at the size of the protein or the shape of it, it's like a rod shape kind of thing.
Mm-hmm.
So yeah.
Okay. All right. Let's see. So, okay, yeah, this is a question I had. How how do you determine you kind of talked about doing the crowded surface, or I guess the concentrated surface versus the dilute surface, and how that affected
Mhm.
the stability and all that. How do you determine that intermolecular distance?
Ah, that's a good question. Um, so what we do is we can measure by cyclic voltammetry the area of the gold
Mm-hmm.
surface that we have before we functionalize it, and then we can measure by cyclic voltammetry at the redox potential of methylene blue. We can measure the number of methylene blue molecules that we have, and we know we have one methylene blue per molecule, and then we assume that they're sparsely distributed, which they're
Mm-hmm.
probably not.
Sure.
Uh, but that's the only metric we can find at the moment.
Okay.
Um, so it's essentially, once you have the number of molecules and the area of gold, you just divide and calculate the packing density and the inverse of the packing density. You can assume a geometry like circles or squares. It doesn't really change.
Yeah.
And then you just calculate intermolecular distance.
Okay, interesting. Yeah, that's that. I guess that makes sense because that was sort of what my question was coming from. Was unless you cover the
For,
whole surface, you won't know for sure. But that's a good way to estimate.
hmm.
Um, I also wanted
Or
to. Oh, sorry. Go ahead.
for DNA, some people have used RuHex in the past, for example.
Ah, okay.
But here RuHex won't intercalate. So, but that luckily, conveniently, we have the methylene blue, so we can use that to quantify.
Nice. Okay. Have you tried to artificially? So you talked about how the surface, the terminal group of the monolayer could affected the stability depending on
Mhm.
what molecule you added. Have you tried to do anything yet with artificially enhancing the stability? Excuse me. Try that again. Have you tried to artificially enhance the stabilization of the proteins via modifying
We
the SAM?
Yeah, we're working on that. We've
Okay.
tried a few monolayers. Not all of them work.
Mm-hmm.
We've tried. Not all of them give you reversible denaturations either.
Okay.
I suspect I've done experiments with, for example, methyl terminated, and those didn't work. We've tried also biomimetic phosphatidylcholine terminated monolayers. Those didn't work either. The carboxylate seems to work, but it's not the most stable monolayer because of electrostatic repulsion in between the carboxylate groups. So that yes, it's easy to change the monolayer. It's not easy to find one that works.
Yeah.
And I suspect we're already starting with one of the best compatible monolayers within the hydroxyl. That's why they're using it for sensors. So the barrier to find something else that works, it's high. Yeah.
Okay.
But that's an active line of research in the lab. It's a very good question.
Awesome. Good. I figured that it probably was, but I was curious. Okay. And the last question we have right now, and I'll go ahead and say if in the chat, if you got any more, go ahead and get them in now. Do you see a pH effect on the stability of the surface-bound proteins?
We haven't looked into that yet because we do also see a pH effect on the methylene blue, because the redox transfer of the methylene blue is coupled to a two proton transfer. So we have, as you can see here, we looked here. So we looked at our peak current. It's also convoluted by the electron transfer rates. So you want to have two rates of electron transfer that are distinct enough. And when you add the pH, those changes. So we know that for protein at 600 hertz, we've done all the controls that we know that we're looking at the the folding thermodynamics of the protein. But if now the rates of electron transfer change because you're changing, for example, the pH, and you're having intrinsic changes in the rate of electron transfer of the methylene blue, then you might have to re-optimize the system and find the new frequencies that are giving you the thermodynamic information. So we that's something we want to do, but it's not so easy to do because it will require some redoing of the controls and re-exploring where is the optimal range to measure that. That would be potentially interesting because then you're changing also the potential at which you're sampling your methylene blue. So you're changing the potential on the surface. So that should be informing you would be able to study electrostatic effects on a different regime of your surface, of your potential, a different potential on your surface. So that it's potentially very interesting. And
very much so.
Yeah.
So I guess I'll add one to finish this off, unless somebody else adds another question, and it may be this was more kind of an open-ended thing. Can this technique be expanded to? Let me think how I want to phrase this. What are the limitations on it in terms of what proteins you can look at?
That's something we're also exploring now. So I suspect that at some point the size of the protein is going to be a limitation. The largest protein I tried, it's 350 amino acids, and that was not reversible on the surface. It irreversibly adsorbs on the surface. So I suspect that at some point, if you add a lot of amino acids, you're gonna favor the equilibrium towards the hydrophobic interactions with the surface, and it's gonna irreversibly stick to it. So I suspect it's gonna be system dependent. Second, I suspect there's gonna be a size limit to the size of the protein that we can put on the surface. Another protein one of my colleagues is working with in the lab, I think it's about 100 amino acids, and that seems to work. So I would say somewhere in between, probably that's where the limit is.
Okay, this is very cool. So I think I'll give one last chance for anybody to ask any final questions. But I think we're going to be done here. But I want to thank you again. This was I do, like I said, I'm chemical engineering. I do nothing biologically related at all. So this was absolutely fascinating. You did a wonderful job explaining the background of it, as well as sort of showing off the really interesting work that you guys are doing. This is a great talk. Really, really fantastic. And
thank you very much, and also again, congratulations for putting this series of talks together. I think it's a very, very good idea to keep us scientists connected in a time like this. and I wanna thank the audience too, because the questions were some of them were very insightful, very engaging. So thank you very much.
That's very kind of you. Yeah, I want to thank the audience as well. I appreciate you guys coming out to ask the questions, because you guys certainly knew more than I did on this one. If I had to come up with the questions, it would have been a lot shorter. Okay, so I think there's no more questions, so I'll go ahead and say thank you to Gabriel. And if I could get you to hang on for just a second, I'll talk with you real quick after we finish here. But I do want to say thank you to everybody that came out and asked questions, everybody that watched. This will be uploaded to YouTube probably later tonight. Usually takes a little bit of time to process, but we'll see. There will not be a stream next week. Next week is my 10th anniversary with my wife, so we'll be doing something not here. But we should be resuming the week after that. So if you follow me on Twitter, or follow it at Meekins Lab, you'll see it there. Otherwise, feel free to join the Discord. Everybody is welcome, undergraduate, graduate, professor of any level, postdocs as well. And I think that's it for tonight. And so I'll say thank you one more time to Dr. Ortega. Again, it was a fantastic talk. And with that, I will say have a good weekend, everyone.